Hello, everybody, and welcome to the Molecular Genetics and Biotechnology Seminars, MGBS 2024. These seminars are organized by the Department of Genetics, Faculty of Agriculture, Asyut University, Egypt. I am very happy to present this interesting talk. This talk is going to be presented by Dr. Abdelaal Shehata. Dr. Abdelaal Shehata is graduated from Department of Plant Pathology, Asyut University in 2016. Then he received an internship for 2 months at Department of Cell Biology, Lund University at Sweden. And now he is a Fulbright master student and a research assistant at Department of Entomology and Plant Pathology, College of Agriculture, Urban University at United States of America. He's going to present. He's working on plant viruses, and his thesis is focusing on sequencing of 2 viruses from Alabama, and to localize the viral proteins and the host in the host of the plant cells. And he is willing to understand more about these viruses' function and their host interactions. And to know more about his presentation and his work, he's going to present his work now. And yeah, please, you can start, Dr. Abdelaal, your presentation. Thank you very much.
Yeah, I would like to start by saying thank you so much, Dr. Amir, for your introduction. And thank you so much for the MEGBS, because I'm always happy to be back to the MEGBS and present every year. It just gives me some kind of different feelings, because I'm sharing my research with my home country. So I hope that you will get the benefit you're looking for from this presentation, and I'm looking forward to your questions at the end. Today, I'll be presenting to you part of my research that I have been conducting in Auburn since 2022. And today's talk is titled basically about protein localization of soybean vein necrosis virus in the plant cells. And this virus is called Orthotus boviruses, which belong to this genus. And I will explain in the next slides what that means exactly. So let's start by talking about the importance of soybean. Soybean is a very important crop, not just in Alabama or the US, but actually worldwide. It is used for so many different uses as textiles, biofuels, and also food source. When it comes to Alabama, which is the state where I live and where I did this research, Alabama planted a lot of soybean in 2022. It is actually the fourth most important crop being planted in our state. The production in 2022 of this crop was 14.5 million bushels, which is equal to 870 million pounds of soybean. This resulted in a value of $206 million. So we're talking about one crop in 1 year produced $0.26 billion to the US economy. So it's very important, as you can tell. Unfortunately, soybean in Alabama is being subjected to a virus called soybean vein necrosis virus, or SVNV. So from now over, I'll be using SVNV to refer to this virus. This virus is infecting soybean in Alabama, and it was first reported in Tennessee in 2008 by Dr. Xenotakis. And in Alabama, it was first reported in 2012 by Dr. Sakura. This virus symptoms, as you can see here in the bottom, the symptoms appears between the veins of the infected leaves, and that's why it's called vein necrosis. And the necrosis is basically batches of dead cells that appears between the veins of the infected leaves. And these necrotic spots usually take this dark color or brown color. The distribution of this virus in the US, as you can see, so far it's found in 16 different states. Those states that are highlighted with orange, they reported the virus there. And this happened since 2008. The host range of this virus is actually very interesting because it's only able to infect 10 plant species so far. I apologize. So it's only able to infect 10 plants so far. And this is interesting because most Orthotus boviruses have a very wide host range, which means that they can infect so many plants. But this virus, until now, it's only infecting 10 plant species. And we will discuss in the next slides or in the results why that could be the reason. So let's talk about the genome organization of this virus. So this virus belongs to a genus, as I mentioned, called Arthrotasbovirus. This genus have so many plant viruses, but this genus as well belongs to a bigger family called Banyaviridae. This family contains more than 350 different virus species. Some of them are infecting plants, some of them are infecting animals. And that's why these viruses are very interesting. And that's because they can infect plants and an insect, which are two different systems. So this means that the virus can replicate in two different systems and they can cross kingdoms, which is very interesting for us. But thankfully, the virus that I'm working on does not infect animals at all, so I'm safe. It just infecting plants. So the genome organization of this virus and all the orthoviruses, their genome is composed basically of three main segments. The S segment, which stands for small, the M segment, and it stands for medium, and then finally the large segment, and it stands for L. So on the S segment, we have two main proteins. This first one called NSS protein, and it stands for non-structural silencing protein, and it's from its name, as you can tell. Actually, the change in the slide, it does that automatically, so I'm sorry for that. But the NSS protein is called the silencin, which means that the virus uses this protein so it can silence the plant response. So basically, it's like silencing the plant defense mechanism so the virus can be successful in its replication. And then the second protein is called the nucleocapsid protein. And as you can see, this N, and it's found in S segment as well. And then the nucleocapsid protein is very important because it does two main things. It builds this icosahedral shape around the virus to protect the virus genome from being degraded in the plant cells. And then also it wraps around the RNA of the virus, forming something called the ribonucleocapsid. And this is also very important because it helps the virus from being degraded. And on M segment, we have the NSM protein, which is the movement protein. And from its name, you can tell that the virus uses this protein so it can move from cell to another cell. And the mechanism that virus can move from cell to another cell is through something called plasmodesmata. And plasmodesmata are very small tunnels between the plant cells. They connect plant cells together. And then the virus have used this protein so it can stretch these plasmodesmata, allowing the virus to move through the plant cells. Also, we have two main important proteins here called the glycoproteins, and they're divided into GN and GC protein. The glycoproteins are very, very important. They are found in the outer shell of the virus. So here are the glycoproteins GN and GC, and they're very important because they work as attachment between that helps the virus to be transmitted by the insects, as I will explain in the next slide. And then finally, the L segment, which encodes to one protein called the RNA-dependent RNA polymerase, which help the virus to replicate in the plant cells. So let's talk about how this virus is being transmitted. So as any plant viruses, SBNV or soybean vein necrosis virus need an insect to transmit this virus. And in our case, thrips, which are very tiny insects, belong to an order called Thysanoptera. These thrips are able to transmit this virus in something called persistent propagative manner or transmission. What did that basically mean? I will explain it through the life cycle of thrips. So if we looked at the life cycle of thrips, they start the life cycle basically as egg, and then the eggs will hatch as larvae. And then the larvae has two different stages. The first one called the first instar stage, and then the second instar stage. After that, they pupate in the soil. As you can see, they go under the soil and then they pupate until they emerge as adults. So what does it mean by persistent propagative manner? Persistent means that the virus can be transmitted by the thrips for their entire lifespans. But this has to happen before something else that needs to happen. First, these larvae here, they need to feed on infected plants before they pupate in the soil. So if they went for pupation before this stage feed on infected plants, they will never transmit the virus, even if the adult fed on infected plants. And it's quite interesting because we think that during their pupation, during the larval stage, they feed on infected plants, as you can see here. So here's the larvae feed on infected plants and taking the virus from the plant cell, from the plant tissues. See, here are the particles of the virus. And then the thrips take the virus through the saliva, and then the virus move to their foregut, and then midgut and hindgut. And through their pupation, the virus uses this period because the insects are sitting there, they're doing nothing, they're not active, they're not feeding. They're just sitting here. And then the virus used this period so it can replicate and reach higher amounts and higher levels in the insect bodies. And then also during this period, the virus moves from their guts to their salivary glands. And this is very important because once the adults emerge, they will feed on infected plants. And as you know, when insects feed on plant, they produce something called saliva. So when they produce saliva that will help them to digest plant tissues, the virus is being released in the saliva again, which is documented here by this blue virus particles. So as I mentioned, the virus has to be acquired during the larva stage, or it will never be transmitted by the adults. So here I'm talking about the significance of this research and why it's important, why am I studying SBNV. So SBNV, as I mentioned, it is infecting soybean, which is a very important crop, and it's enough reasons that's why we study this virus. But actually, there are so many more reasons that why we study this virus. This virus was found in 16 different states just between 2009 and 2015, which indicate that the virus is actually spreading more and more. I have read some studies that says that right now in 2024, the virus is found more in more than 22 different states, which also means that the virus is spreading more and it's becoming more severe and more dangerous. And in Alabama, the virus symptoms have increased from 31% to 82% only in a 3-year period. But it is also it's not the same every year. Sometimes we will have a higher year for the virus, like a higher incidence. Maybe the next year would be low incidence, and that's based on the weather conditions. But this was documented in just 3 years, and it's also worth it. And that's why we studied this virus. But most importantly, this virus, since it was discovered in 2009 or 2008, it has never been localized in the plant cells. And we don't know any idea about how this virus functions in the plant cells. We just know that it's infects soybean and it causes more damage, but we don't know how it's function. And as long as we don't understand how it functions, we cannot manage it because we don't understand how it works. And that's why I did this study. It's called localization. So what is this protein localization and why it's important? So protein localization is basically a process where we fuse the proteins of interest. So the proteins of interest can be virus protein, can be bacterial proteins, fungal proteins, any proteins. So we take this protein and then we fuse it with a plasmid that has a fluorescent protein. And then after that, we take this plasmid and then we insert it in our plant or in the host that we are trying to study the virus in. In my case, I will be putting the proteins in the plant cells. So when I put the proteins in the plant cells, they're going to be expressed as an actual protein. And then the protein is going to be localized in some spot. Maybe it will be in the nucleus, maybe it will be just in the cytoplasm, maybe it will be in the plasmodesmata as here. It's going to be in somewhere, and I'm going to see this under the microscope, right? And then this will give me an idea how the virus is functioning. Because if it's localized in the nucleus, I can have an idea like why it's localized in the nucleus, or why it's in the cytoplasm, or why it's in the Golgi. It's going to give me an idea how the virus is functioning. And then from this step, we can start developing management methods and then how we can prevent the virus from infecting these cells, right? So basically, my hypothesis is that once I do the protein localization, this will help me to answer a question, which is where each of SBNV proteins localize in the host cell. Once I know the answer for this question, I can have a better understanding, or I can answer also this question, which is how the SBNV infection happens in the plant cells, right? And then finally, we can develop management methods to this virus. So here are my hypotheses. And then my objective for this study today is to clone and localize and co-localize SBNV proteins in the plant cells to determine where these proteins are in the cell in comparison to other orthotasboviruses. So here are the methods that I use. So we use the reference genome of SBNV, which has come from Tennessee. And we did this because at that point, we did not have SBNV isolated from Alabama. So we used the reference genome and then we amplified the 5 genes, which are NSS protein, N protein, NSM protein, and then GN and GC, which are the glycoproteins. So I have designed specific primers to amplify each of these genes, and I ran the PCR, and then I ran the gel, and as you can see, here are the sizes of each one of them. After that, I took these PCR products, and then I purified them, and then I cloned them in the p-enter vector. And in the p-enter vector, your forward primer has to have something called CACC, which are complementary to the p-enter sequence. After that, I sent them for sequencing to make sure that the sequence was good and I don't have stop codons or like bad sequence. So I sequenced them, and after I make sure that all the sequences were perfect, I finally used them and I moved them in the p-side vector. So the p-side vector is called the destination vector. This is the final vector that I will be using to insert my proteins in the plant cells. And here is the process. So this is the entry clone, and this is after I cloned the genes and I sent them for sequencing, and the sequences came to me and I analyzed it, and it was good. And then I ran something called the LR reaction. The LR reaction allows me to move the gene of interest from the entry clone or the donor vector to the destination vector. My destination vector that I use, as I mentioned, called p-side 2Na GFP. So p-side stands for plasmid, stable, integrated, transient expression, which is a very stable vector under the plant cells. It doesn't get degraded or something. It's very stable. And 2Na means that it was fused to the N-terminal of the protein, or the N-terminal of GFB in that case. And then GFB stands for green fluorescent protein. And this is what you can see here. So here's GFB. And the plant or the virus protein will be fused to the N-terminal of the GFB. So after that, this will be like put in the plant cells, and then it will be expressed as a protein. So the protein of interest will be attached to the GFB, but actually we're not going to see the protein of interest. We're not going to see the virus protein. What we're going to see is the GFB, which is going to be as green. Okay. And that's why we use GFB, because if we didn't use any fluorescent protein, we can't see the virus proteins, right? And the virus protein actually in a plant cell is invisible, doesn't have color. I just colored it here in purple so you can imagine what am I doing, okay? So, and here's the process where I basically did it. So I got the clones and p-side, all of them, and then finally I moved them into agrobacterium. So agrobacterium is used because in nature, agrobacterium infect the plants and they insert their plasmid into the nucleus. And that's what we want. So we want a bacteria that's going to deliver our plasmid into the plant nucleus. Because if the plasmid did not go to the plant nucleus, it's DNA, right? So it has to be in the nucleus because the nucleus has the DNA polymerase. So we need the plasmid to be in the nucleus. So we found that agrobacterium does this in nature. So we used it in the lab. We made it as competent cells first, and then we give them the plasmid. And then after that, we grow agrobacterium for 5 days on petri dishes plated with good media. And then once the bacteria grows, we can streak it, we make the OD solution, and then infiltrate it in the plant. So that's what I was doing here. So basically, I grow the bacteria, and then I make the OD. So each bacteria has, like if you looked into one cell, this cell has the plasmid, right? And then the plasmid has GFB, and then the protein that I want to test, or I want to see where it's going to be in the cell. And then I'm going to perform the infiltration, which using the plants called Nicotina benzimiana, or we can actually call it benth, as we call it in the lab. And then the reason we use benths is because we have 2 reasons. First of all, it's very easy to infiltrate. This is infiltration. Infiltration using a syringe, and then take some from the bacteria solution and then force it to go into the plant cell or to the plant tissues, but without using needles. We don't use needles here. So we don't inject the virus. We basically infiltrate the virus. This is different. So we use benths for 2 reasons, as I mentioned, because it's easy to infiltrate and because it's actually, it can be infected by this virus in nature. So we cannot use a plant that is not infected by the virus because the virus is not going to work in this plant. So it has to be able to infect by the virus. And then we're going to wait. So as I said, the bacteria will deliver this plasmid into the plant nucleus here. And then this will take it 2 days, basically, or 48 hours. This is 48 hours post-infiltration or days post-infiltration, as you will see in the next slides. But this will take it 2 days until the protein is being expressed and an actual protein. And after that, I'll be taking some discs from the plant that I infiltrated and then make slides like that and then put it under the microscope. And then I'll be able to see the virus protein. So let's get into the results. First, I will be showing you the localization results. In these results, I will be just looking into one protein every time, okay? So I infiltrated just singular protein every time. So first, we actually have the NSS protein. The NSS protein that was localized first, and as you can tell, So first, before I show you the results, you have to know that the first panel here shows the protein of interest tagged with GFB. So the green here refers to the virus protein, which is NSS. This panel here is a plant called RBH2B. And this means that this plant is a transgenic plant and its nucleus glow as red. So what you see here in red is the plant nucleus. And then finally, this is an overlay between the GFB and the RFB, as you can see. So I just overlaid them together. So in the first panel here, you see that the NSS protein is localized into the cell periphery or the cell wall, and also localized in the plant nucleus, as you can tell here. Also, this protein was aggregated in clusters, as you can see. These are aggregates from this gene. And when you overlay it, it shows you the nuclear localization. This dark spot in the middle is something called the nucleolus, and nucleolus has the ribosomal RNA. So I was not expecting to get nuclear localization for this gene because the other localized arthrotopoviruses did not show nuclear localization except for one virus that did this for its NSS. So because I was not expecting these results, I wanted to look for something called NLS domains. What are the NLS domains? It stands for nuclear localization signals. And it basically means if you have a protein, you can just put it in an app or like a program and it's going to analyze it for you and it's going to give you if this protein has any nuclear domains or not. If the protein have nuclear domains, it can improve or can support that, yes, maybe nuclear localization is something possible for this protein. So I looked for that and I actually found that this protein has nuclear domain. As you can see here, this one in red is a domain that was detected in this protein. And it has a score of 4.8. And it's also called bipartite. When I looked this also, this app here tells you like if you have a score of 7, it's highly possible that's going to be just in the nucleus. And the more you go down, the more you have maybe less nuclear localization. But basically, this score was reported to be, it could be both nuclear and cytoplasmic. And that's what I saw. I saw cytoplasmic localization and nuclear localization. The first or the second gene was the nucleocapsid, and that was even more interesting. The nucleocapsid protein of this virus also localized into the cell periphery and did not aggregate and localized in the nucleus. This one was more seen in the nucleus compared to this one. And you see this by the change in the color. When you overlay red with green, it gives you yellow, right? This one here gives you a little bit of yellow, but not as this one. So the nucleocapsid protein of this virus was also localized in the nucleus. And when I looked if this protein have any nuclear domains, it actually has 3, not just 1. One of them has a score of 5, which is interesting because it was the highest that was observed in this protein. And then NSM protein was even way more interesting. And if you ask me why, because this protein killed the cells. So every time I infiltrated this protein into the plant cells, it did not localize normally. Like I didn't see normal localization. Every time I see the cells are turning to death. And you can tell this is not actually like normal localization. And if you ask me why, because if you looked into the RFP, which is your control plant, it showed the same behavior here. You see? This is not different. This is the same as this. It's just different color. So this means that this pattern is not related to the virus protein, NSM. It's actually the protein is killing the cells. And these were the symptoms that were observed in the infiltrated leaves. As you can see here, we get necrosis, or basically cell death. So NSM protein, which is the movement protein, killed the cells and caused cell deaths on them. And then we have GN, which is the first glycoprotein, and this glycoprotein localized into the cell periphery and around the nucleus. This was more around the nucleus, as you can see here, just around the nucleus. When I looked for NLS domains, it does actually have 2 domains. And then the GC proteins also localized to the cell periphery or the cell membrane and also around the nucleus, as you can see. So we just surrounded the nucleus, not inside of it. Also, it did show nuclear domains. It has actually 5 nuclear domains. Finally, I used the free GFB, which is just a plasmid that doesn't have any proteins in it as a localization control or negative control. So the free GFB, as it mentioned by all studies, it localizes to the cell periphery and the nucleus. That's what you can see here. So this is my control, and it looks different than my treatments or my results. After that, I will get into the co-localization. And in this here, I want you to focus because I will be localizing two proteins from the same virus every time. So I will be putting two different proteins together in the same time. And this is important because I want to test if the presence of one protein will change the localization of the other proteins or not. This is what I want to know because in nature, the virus, like in the infection, in the natural infection or in the real infection, the virus is being all together, right? All the proteins of the virus are being together in a cell. So when you see individual localization, which I just showed you, it could be different when the virus is present together, right? Because we have more proteins, they could interact with each other, they could influence or impact each other localization. And also, I want you to imagine if I have two proteins and I'm going to put them together in the same cell, this will require that I'm going to attach each one of them on different fluorescent protein, right? Because if I put both of them on green, I can't tell which one is affecting the other one, right? So in these results, I will be attaching one protein with GFP, which is green, and then the second protein will be in red, which is called RFB or RB. Okay, so for the co-localization results, as you can see here, this is the overlay, okay? This one here is the first protein attached with GFP, and this one here is the N protein attached with RFB. So here it referred to the N protein with RFB, which is red, but this one here referred to the NSS protein attached with GFP. The plant that I used in this case was wild type plant, which means it does not fluoresce in any color normally. It's just black. So in the first panel here, I co-localized NSS with N. And what I saw is that their nuclear localization was exactly the same. So NSS also localized in the nucleus, and then N also localized in the nucleus. It wasn't different. Both of them, they showed the same localization. And for GN, I used GNS. This is called soluble version. Maybe I didn't mention that before, but GN as a protein, it has something called the transmembrane domain. And the transmembrane domain takes the protein after it's been made and stick it into the plant membrane, and this will make it hard for us to image it. So I actually co-localized GN full length, but it did not fluorescent. So I use a soluble version, which means that I cut the TM out of it. So I used the soluble version of GN and also GC, and I co-localized them with RB, NRB, and then NRB as well here. They both showed similar localization, as you can tell. This one here is the same as NRB, and when you co-localize them, they turn to yellow. So all the spots that actually show yellow color, this confirm the co-localization, which means they behave the same way in this spot. So also GC showed the same co-localization as N. And then here I co-localized GN and GNC, which are full lengths here, they're not soluble version, with NSS and NSS. They also show the same localization. As you can tell, this one in green showed the same one in red, and when you overlay them, they turn yellow. This one here also showed the same co-localization with NSS. And then finally, I co-localized GN, or here GN with GC, and then they also co-localized the same. However, the new co-localization was less seen in the GN and RB, but it was more privileged to see it in the GC soluble version. So these co-localization give this idea about if any protein will change the co-localization of the other or not. That's actually what I saw here. So this was very interesting because I showed you this one before. This one was the first one that I showed you here, right? So I did a time course. A time course means that I will be imaging every day for 7 days. So I was imaging this protein together every day until 6 days. And that's because I saw that they changed by the time. So at 2 days post-infiltration, they were very soluble. They localized to the cell periphery and to the nucleus, as you can see here, right? But at 3 days, they did this behavior. So I saw that NSS and GFP completely aggregated in this big cluster, okay? And then also NRB did the same. And that's why when you co-localize them, they turn to yellow. They didn't do this when they were localized individually. And this is something interesting. Also, I saw that the NSS protein in GFB, it also aggregates in punctuated clusters, like very round clusters. And they are taking a shape of something like the nucleus. I can't tell if it's nucleus or not, I can't confirm. I'm just thinking that it could be the nucleus. and that's because it's taken the shape of the nucleus. And when you look into the NRP, you see these like in the same spot where you have punctuated protein aggregation from NSS, you see them as black halos, which mean that the virus or the protein is existing in these spots. But at 6 days, it was even interesting and funny as well. It's funny because, as you can see here, NSS actually aggregated in complete circles, but it actually making a scary face, if you can tell here. This one looks very scary. If you like rotate it in some angle, you will see like a smiley. I don't know if it's smiley or scary, but it looks funny to me. But basically, NSS was aggregated in clusters, in very round clusters, and NRB was surrounding the protein aggregation of NSS. This was seen many times. Also is here, as you can hear. There is another face down here looking at you, and then also the N also was localizing here. And I used DABI. It's a stain that you infiltrate before imaging, and it shows you the nucleus. So it showed the nucleus in a blue color, and this could be the nucleus, this one here. So what I'm saying that NSS in the presence of N, this one here, at 6 days post-infiltration, it aggregate in the nucleus, as you can see here. And N is surrounding the NSS from all sides. We don't know, maybe there is an association between N and NSS, and maybe they interact in the real infection. We don't know, but this actually suggests that they could interact because they were found together, very close together, surrounding each other. So let's talk about the discussion. Okay, so before I did this study, I actually looked into this phylogeny tree. This phylogeny tree here shows us all the sequenced orthotusboviruses. And here I want you just to focus on these 4 viruses that are circled in red. This one here called IYSV, or ambitious Irish yellow spot virus. And then this one here, Capsaicin chlorosis virus, which is CACV. And this one here is INSV, which is ambitious necrotic spot virus. And then finally, TSWV, which is the most well-studied orthotusbovirus. It stands for tomato spotted wilt virus. What I want you to see here is that SBNV was found distinct or far away from all these viruses. It was found in a different clade, just with one virus called B necrotic mosaic virus. This gives us idea that SBNV is actually distinct. It's different. It's not like other orthotusboviruses. And this also gives us idea why the proteins of this virus were different. So for the comparison here, because in my research, I said that I'm going to compare the localization of SBNV, which I'm going to show you here, with other characterized orthotusboviruses. And I only selected Capsaicin chlorosis and TSWV because these 2 viruses show they were so different from each other, and they show different, like 2 different groups in the localization of these group of viruses. So for Capsaicin chlorosis, for its NSS protein, it was the first orthotusboviruses that they said that it's NSS in the nucleus. But for TSWV, it was just cell periphery and around the nucleus. And actually, our SBNV does the same as this one. So it localizes into the cell periphery and to the cell nucleus, as you can see here. For N protein, it has never been shown to localize in the nucleus in all orthotusboviruses. It always cell periphery and aggregates in the cytoplasm. This one here was just around the nucleus, not in it. They didn't say in their study or in their paper, they didn't say that the nucleocapsid was in the nucleus. But for our N, it was actually in the nucleus, as you can see here. And this is the first documentation for the nucleocapsid protein of any orthotusbovirus to be in the nucleus. And this is very, very important. It could change our idea about maybe orthotusboviruses, or at least about this virus itself. The NSM protein for all orthotusboviruses always localize normally in the plasmodesmata. As you can see here, I mentioned to you before what are the plasmodesmata. They are small tunnels that connecting the plant cells together. And NSM of tusboviruses, as Capsaicin chlorosis and TSWV, it shows localization in these plasmodesmata, which is normal, right? Because the virus need to be moved from cell to another cell, and NSM does that. But for our SBNV, its NSM is not functioning very well. It kills the cells and it doesn't localize normally. For the glycoproteins, finally, they both localize in a cell membrane and around the nucleus, matching with TZOV more than SVNV. Sorry, more than capsum chlorosis. So as you can tell here, I think, or I feel that SVV is more related to capsum chlorosis than TZOV because it matches with one protein at least here. So for my discussion also, I would like to mention that NSM protein, which causes cell death, it could be the reason why this virus has very narrow host range. If you remember in the introduction, I mentioned that this virus is only able to infect 10 plant species so far. And this is interesting because most orthoviruses have a very wide host range. For example, TSWV or tomato spot wilt virus, it's able to infect more than 1,000 plant species. And I think this is due to the NSM protein. Because in SVNV, the NSM protein killed the cells. As you can see, when I infiltrated the proteins in the plant cells, and it was just one protein, which was the NSM, it causes the cell death and it killed the cells. And this is the symptom that we see in the real infection. These are symptoms that were collected from Alabama field in last year. So I think that NSM protein is causing the symptoms of this virus, and it's responsible for the very narrow host range. Because when it killed the cells, thrips, which are the vectors, will not be able to feed in these dead area because it's dead. So insect will not feed here. They will actually go to like a healthy tissue so they can feed. And then this also means that the virus is not able to be moved between the cells because NSM is not functioning very well. So what is the thing that's going to come and move the virus from cell to another cell? There's nothing because NSM is not working very well. So I think that the virus is basically trapping itself in these dead tissues, which is limiting the virus ability to be transmitted by insect because they're stuck here, right? Also from the co-localization that we saw that the co-localization looks the same for all of them except for NSS and N protein. They co-localize differently. They show the aggregation in the cytoplasm or in the nucleus, and also N was always found surrounding NSS, and this is interesting, and this could mean that they interact. This doesn't give us idea or doesn't confirm if they interact or not, but to test the interaction, we need to use something called the pull-down assay. And this is what I'm going to be using in the next research studies in the future. I'll be using the pull-down so I can see if any of these proteins will interact with each other or not. And this is very important as well. So as a summary for this research, I want you to think here, or actually to answer this question, what is the end game goal for viruses? What are they looking for? What is their end game goal? Like, what are they looking for? If you think about it, viruses are so selfish, right? Yeah, they go into the plant cells, they say, let's hit it and then meet our goals. Do you want just two things, to improve and replicate more? They don't think about the plants, but also we have to distinguish between a smart virus or let's say successful virus and unsuccessful virus. Successful virus wants to keep the plant alive, at least for a little bit. Why? Because you need to replicate, you need to increase your numbers before the insects come and take you and then transmit you to another plant, right? But unsuccessful virus or a failure virus would kill the plant right away. And by that, it limits its chance to be transmitted because it killed the plant. So it trapped itself into this dead plant. And then the insect will not feed on the tissues, right? They need healthy tissues. So we have this association clearly here between TSWV and SVNV. But given the fact that TSWV was found 110 years ago since it was discovered, but this one only had 15 years. So the 1 century, the 100 years ago, gave this virus the ability or the enough time to mutate, right? Like I suspect that this virus, when it was first reported, at least maybe it would have like very few host range, but now it's able to infect 1,000 plant species. That's a huge number of plants. Okay, these 10,000 plant species belong to different 70 plant family species. I'm sorry, I said it wrong. So this 1,000 plant species, they belong to 70 different plant families, which means that this virus can infect everything basically. But given the fact that this virus has been around for a very long time, it had enough time to mutate and it adjusts its genome. Now, TSLV is only not killing the plant, it's just keeping the plant alive, causing the perfect symptoms, which are called yellow ring spots, but it doesn't kill the plant. And by that, it keeps the plant alive, the virus can replicate more, and then can replicate and mutate until the insect come and pick it up and then move it to another plant. But for our SBNV, it's only been around for 15 years. Now it's killing the cells. It's not allowing the insect to come and transmit it. And then, until now, it only has 10 plant species. But also there is something very important about these viruses. These viruses have something interesting called genome reassortment. What is genome reassortment? This means that if this if this virus have the chance to meet with this virus in the same plant or the same vector, they can share some genes between them. I can like give this virus a gene and then take a gene from him in returns. This is called reassortment or exchange, genome exchange. So this actually can happen easily between these two viruses in Alabama. Why? Because we have two reasons. The first reason is both these viruses are transmitted by the same insect species, which is called the Frankliniella fusca, or the tobacco thrips. They both can be transmitted by the same insect, which means that it's possible that both of them can meet in the same plant or in the same insect in this case, right? Which means that the genome reassortment can happen in the insect. Also, we have another reason, which is SVNV in Alabama is mainly infecting soybean, okay? And TSOV is mainly infecting peanut, but also this virus was reported in soybean in Alabama 11 years ago. It was also reported in soybean in Georgia, which is very close to us. It's our neighboring state. So we have two reasons that both these viruses can meet in the insect or can meet also in the same plant. This gives us idea that these viruses can meet together and reassort, and then maybe SVNV will get the movement protein from this virus, and then this virus can actually spread widely and infect so many plant species. And in that case, we will be dealing with two destructive viruses instead of one. TSOV is already very destructive. It costs millions of dollars every single year as yield loss. SVNV doesn't cause a huge yield loss until now, but if this virus was able to meet with this virus and reassort with it, SVNV will be very destructive. And of course, we don't want this to happen. It's going to be very sad for everyone, for the farmers and for the people. So we don't want this to happen. And we need to focus our research and our funds to fund these projects and try to prevent this before it happens. At least to try to understand how the genome reassortment happened so we can maybe prevent it or stop it. That's it for me today. Thank you so much for listening. My acknowledgement, of course, I would like to thank our lab, Dr. Martin, our PI, and everyone in our lab, including the undergrads and the graduate students. Thank you so much for helping me. And also, I would like to thank that this work was funded by our university, and also it was funded by the Agricultural Research Enhancement, Exploration, and Development. Oh. 1 second, please. And then also, it was funded by the National Peanut Board. Thank you so much for your listening, and I'm ready to take up your questions.
Thank you very much, Abdelaal, for this very nice talk. I'm sure there's a lot of questions. You can ask the questions in the on the chatting room. Actually, I have a question for you, Abdelaal.
Yes, please.
So why did you perform your research on a different plant than soybean? Why you didn't use soybean directly to localize these different proteins?
Yeah, that's a really good question. And it's because soybean sucks. Soybean is really hard to infiltrate. You can't infiltrate soybean. We tried so many times to infiltrate soybean, but you cannot infiltrate it ever because the leaves are very thick and the virus cannot go through them. It's very hard to infiltrate it in soybean. But also plant, I'm just gonna keep answering your question. Also, as I mentioned, beans or the plant that I use for infiltration, it also can be infected by the virus in nature, which means the virus should behave similarly in soybean and in beans. It should show the same thing. So it shouldn't be different.
And the virus is only affecting the leaves, or there's other type of tissues can be infected by this virus?
Yeah, so this virus can infect leaves mainly, but also can infect the soybean seeds. So
do you think that the localization will be affected by the type of the tissue? So you tested on why don't you think about it may behave in a different way in the leaves or in the roots or in the seeds? Do you think that they will have the same behavior?
That could be possible. That could be possible that they behave differently based on like the tissue, if it's leaf or seeds or stems or flowers. It could be possible, but actually we cannot infiltrate seeds because you can't put the virus in them and see them under the microscope. It's hard to take tissues and put it under the microscope. But I expect that it's going to behave similarly. And then when the virus moved to the seeds, it just moved. I didn't mention that in my research today because it's the limit of time, but the virus is moving to the seeds using another virus. There is another virus is implicated in moving this virus to the seeds. So the virus by itself, it doesn't move to the seeds. It just needs another virus. But it should behave similarly to based on the tissues. It should be the same.
Yeah, and do you think, sorry, I have more than one question. So
Yeah.
do you think
I like
that,
questions.
so you are inserting the plasmid which have the gene of the different proteins. Do you
Yeah.
think that it's the same as what happened in nature? Because I think in nature the virus with all the
Yeah.
proteins is going to infect the cell. So and
Yeah.
do after infection, the virus is like uh, is it's in that same in the it will be in the same structure. I mean that it will be the
Yeah.
whole virus together, or it will split to different proteins. I don't know. So
Yeah.
why do you study every single protein individually?
Yeah, it's because we don't have something called infectious clone. Infectious clone basically means that we put the whole virus on one plasmid, but we can't do that because the virus as whole virus is very large to put on a plasmid. It's more than 15 kb, and we can't put these 15 kb on one plasmid. And also, if you notice here, the virus is found in different segments. You see, these are different three segments, which mean that we have to put every segment on a different vector and then infiltrate them together. This is called infectious clone, and it's very, very hard for these viruses to be made because these viruses have genome called negative sense and ambisense. So on the same segment, you have genes in different directions,
Yeah.
and their genome is even different. It's called negative sense, which means it's not complementary to the mRNA of the plant. So it's really hard to make infectious clone for these viruses. But of course, I agree with you that infiltrating or localizing individual proteins may not be the same when you have a full virus, and that is so true. And that's why I did the co-localization. The co-localization here, it shows you if the proteins, when they're together, they behave differently or not. But when I showed it here, it was basically the same except for these two proteins together, the NSS and N, they behave differently.
Yeah. And why didn't go further for like testing more like more proteins together? So more than two proteins? It's
Yeah.
harder, right?
Also, we also have limits. We have only GFP and RV. We only have two colors, green
Yeah.
and red.
Yeah. And
We all, you
you
know,
don't
in
think
research we
that
have limits.
tagging these proteins with like GFP is not going to affect the function of the protein?
No, it shouldn't, and that's why we use free
Negative.
GFP as
Yeah.
like a negative control. You see, negative control localized differently.
Yeah. And so my last question.
Yeah, of
So
course.
what is your recommendation? So now you did a great job. You know the location of each protein.
Yeah,
How you are going to use it to like apply
yeah.
resistance to this virus?
Yeah, so it's a very good question. So far, I mentioned at the beginning that before I do this study, this virus, it has never been localized in any lab around the world. We didn't know any idea. Now we know at least how the virus is functioning in the plant cells. Of course, we don't know exactly how it's functioning because we need to do more studies. So my recommendation is that, of course, I'm going to be continuing doing more studies as pull down and actually, one of my projects for the next future is to develop infectious clone. However, it's very hard, but that's one of my plans. But I'm willing to understand this virus more and more. But for now, my recommendation for the management is basically to manage the insects. Make sure that the insect levels in your field is very low. Use insecticides so the insect cannot be present more, because if the insects are found more, they will transmit the virus more. So far, we don't have, like based on these results, the localization results, we don't have a management methods based on these results yet. And that's because we need more studies because before we say use this pesticide or do that or that. We need to do more research so we can understand how it's functioning. But so far, I can tell you maybe a hypothesis. So as you see that here, yeah, we showed that we have two proteins in the nucleus. NSS and N protein. This gives me idea that maybe this virus need to be in the nucleus for its replication. And if we can prevent that, if we can prevent the virus to go to the nucleus, we can actually stop the infection. And I will be just targeting this nucleocapsid. And if you ask me why, because the nucleocapsid is very important. Without the nucleocapsid, the virus cannot be assembled in one full virus and then infect or move to another cell. Without that, the virus segment will be just lost in a cell. So if we can stop the nucleocapsid from going to the nucleus, this could actually inhibit or prevent the virus from being replicated and transmitted.
Thank you very much, Abdelaal, for this very nice talk.
Yeah, sure.
I don't know
you.
if we have more questions or not. No more questions.
Do you check the chat? There's something in the chat, maybe?
No. Okay, thank you again. And
yeah, sure. Thank you so much for hosting me. It was really
thanks.
my pleasure to be hosted today.
Yeah, me too. We were very pleasure to have you in our seminars. Thank
so
everybody for the nice listening, and see you soon in the upcoming seminar. Goodbye.